So our next speaker, also from Caltech, making this an all-tech afternoon, it's a special thing, was, as a grad student at Rice, was the co-discoverer of C60 fullerenes, with a discovery that was subsequently recognized with the Nobel Prize, of course. At UCLA as a faculty member, he founded a multi-institutional organization called the California Nanosystems Institute and served as its first director. Then moved to Caltech in the Department of Chemistry. He's a fellow of the American Physical Society and a recipient of the Feynman Prize in nanotechnology. And today he'll tell us about systems biology-driven in vitro diagnostic technologies for cancer. Please welcome Jim Heath.
Thank you. First, it's a pleasure to be here. I want to thank Bob for the invitation. It's exciting. I know that the Koch Institute is still a hole in the ground, but it sounds like it has a fantastic future, and I wish you all the best. Just a few disclaimers, but today I'm going to switch gears a little bit from the previous talks and sort of be complementary to them and talk about diagnostics and technologies for diagnostics of cancer. And so let me begin with, well, this is a data from one of my collaborators, Paul Michel, who's a pathologist at UCLA. This is very advanced pathology for, it turns out, brain cancer. Typically, a pathology report for brain cancer is about 1 line long, and it just says there's cancer there. And but what's become recognized over the past several years is with all cancers that, it's like Tolstoy's comment, all happy families are alike and each unhappy family is unhappy in its own way. Cancers have a great diversity. And that diversity is reflected in measurements of their gene and protein expression levels. And so, if you and this is particularly relevant for this disease here, glioblastoma, because basically if you get it, you're going to die. And the way it's normally treated is that once it's diagnosed and a biopsy is taken, a patient's put on a regimen for, there's like 4 possible regimens, put on one for 3 months, monitored, and then if that doesn't work, another one. If that doesn't work, another one. And that's not good if you have like a 4 or 5 month outlook for survival. And so what Paul has developed, and this is a little bit idealized, it's a few years old, but if you take the tissue sample from these patients and you measure mRNAs, proteins, post-translationally modified proteins, and you do the traditional pathology, within the context of this thing here, there must be a pointer here somewhere. I have one that my mother gave me, but I use the other one. Okay, so this is, what's sort of, I guess, the most clinically accepted model of disease. It's a pathway model. And this is meant to be a membrane protein, and so a molecule comes and binds here and triggers a whole cascade of events that, eventually, for example, will send a transcription factor into the nucleus and get the cell to divide or something. And the idea with all these different forms of cancers is that there's a host of different ways to short-circuit this pathway. And so it's like being in this room and the lights are out. And you don't know if it's because the light bulbs are unscrewed or if the light switch is off or if the circuit breaker's off. There's one result. The lights are out. A lot of core reasons behind it. And in this case, one result, you have cancer. A lot of potential core reasons. So you do these various measurements and you set up a sort of decision tree based on these various measurements. And then you can put patients on drugs, and you'll see that each of these patients for these particular inhibitors responds, but that patient, for example, will probably not respond to this drug, and vice versa. And it turns out, even as idealized as this is, as people begin looking deeper into this, things like combination therapies, or it's not just one kind of cancer, it may be actually a couple of different kinds of cancer, things like that are becoming important. And so measurements are becoming important. And so a challenge would be, how could we do these kind of measurements non-invasively, not having to open up and operate on the patient? And the first challenge that you really have to get the biology right, and if you want to do it non-invasively, you're probably going to be looking at the blood because that's the largest and deepest version of the human proteome. And it has sort of a staggering complexity in terms of the numbers of proteins, the dynamic range or the concentration range of those proteins. It makes it an extremely noisy environment. And these pathway models are tremendously limited because, well, let's just take this one protein here. And if you look at the diseases that it's correlated with, here's what the literature pulls up. And so EGFRV3 is a fine cancer marker on a tissue. But if you somehow had a corresponding protein in the blood that was a good cancer marker, it would not tell you much about where the cancer is. It might just tell you you might have cancer. And so you have to have ways to look into the body that give you specificity to let you discriminate between these various diseases. And that's been a real challenge in biomarker discovery. You know, and oftentimes, you know, a biomarker fails because it looks great when you look at 10 brain cancer patients versus 10 healthy patients, but as soon as you throw in all the other cancers and diseases, it falls apart. And so we work with this gentleman, Leroy Hood, who I think has been here a few times. And he's come up with one, what I think is kind of a non-obvious strategy, and one which to a physical scientist is a little more straightforward. The non-obvious one is to do really comprehensive measurements of mRNA expression in every organ in the body, and then ask, well, what is it that makes the ovaries different from the breast, different from the prostate, different from the brain, et cetera? And ask that at the molecular level. And it turns out if you do, what you'll find is that each organ has got a host. Here's an mRNA that's expressed only in the prostate. It's not PSA, it's a different one. And here's all expression level in all the other organs. Here's just a few of these organs. There's a list of about 60 or so now. And you'll find that each of these organs has got, depending on where you cut the baseline off, has got a host of proteins that are only expressed in that organ. And if you look even in healthy patients with fairly low sensitivity mass spectrometry, you can see anywhere between 10 and 20 of those. And probably with a lot of sensitivity, you might be able to up that up to 40 or something. So that means that in the blood, you have the potential to look at 40 or so, 20, 40 proteins that are reflective of the health status of all the individual different organs. It's a lot of measurements, but it's a way of overcoming the signal to noise if you can manage to do those measurements. The other thing, and this is what's more evident to physical scientists, is that as you do measurements as a function of time, over a fairly rapid time, then the noise averages and the signal rises. So here's an example of looking at a tox event. This is also from Lee's lab. It's acetaminophen toxicity in a mouse, looking at on the order of a dozen liver-specific secreted proteins, so looking at it through the blood. And it turns out this whole event is over and done with in about 2 days. But during the course of that, and here's the pathology of the liver as you go through that tox event. But as you go through that event, 4 of these proteins, liver-specific, are upregulated, and another 7 are down. And this actually constitutes a fingerprint of this, I don't know how unique, but it has some unique character to it, of this acetaminophen toxicity. So that's the second strategy. And the third one has to do with actually knowing what the physiological function of these proteins is. So not only do you know what the organ it is from, but you know its biological function. And so as we go from this sort of where medicine has been this sort of single parameter measurements to these pathway measurements, which are somewhat incomplete but still useful, to this kind of a model that looks somewhat like biology hits a windshield at 50 miles an hour. That's actually a model of prostate cancer, and it looks completely unintelligible. But it's not actually, you can extract good information out of it related to the various protein networks. But the point is that you can mine these kind of models for molecules that can tell you answers to questions like this, and what you call an informative diagnosis. But you're talking about doing a lot of measurements, and you're talking about doing them at high frequency. And if any of you are online right now, and I'm sure if you're online, you're not paying attention to what I'm saying. But Google and look up and see what it costs to do a CA-125 blood test or PSA. It's $50, and the hospital is going to charge. And we're talking about doing 1,000 measurements. And so what you have to do for the biological measurements for these proteins is to drop the cost down just like a transistor's dropped from down to, a fraction of a penny per transistor. You have to do that for biological measurements. Or this kind of medicine where we want to see happen is not going to happen. And so if you think about how it's done now, well, you take, actually 5 milliliters of blood is sort of the minimum collection. Guarantee there's a cost there. The patient feels that. You centrifuge to separate plasma from the serum, like this. That generally involves either person or time. And time has a lot of cost associated with it, and I'll get to that. Proteins in a 96-well plate, oftentimes this is diffusion limited. And so if someone runs and just does this as fast as they can, it's going to take a few hours. And oftentimes it's sent off to a lab, and it takes a few days. And so you'd like to take all of this stuff and just drop it all by orders of magnitude. And I think, and we actually, I like to make widgets. And so, but that's not what's called for here. The science is asking you to do something different. You don't really want widgets. You just want to make things really cheap. And in fact, I think you're allowed to use, for these measurements, glass and plastic and really not much else. You need inexpensive reagents. That's going to be the bottleneck very soon. It's very small amounts of tissue or blood. And this enables you to look at mice routinely, for example, as well as finger pricks and things like that. You want to do it quickly, multi-parameter, scalable, minimal human intervention, et cetera. And so we developed, and actually this is not, I think a lot of the stuff that I'm going to talk about has been done piecemeal by other groups, but the integrated effect and how we're using it, I think, is pretty powerful. So a postdoc in my group, Brian Bailey, initially addressed this issue for the glioblastomal problem of how can we, in one platform, condense those measurements of mRNAs and proteins, et cetera, into just one platform. And it turns out the chemistries are not compatible. So they're generally done with different sampling, different allotments. But it turns out this method unifies them all. So you just take the antibody to the protein or the cell you're trying to sort or what have you, and you label it with DNA. We make all of these DNAs orthogonal to each other. And it turns out that combination of them being orthogonal to each other and also being DNA really limits biofouling. And then you can use that type of a platform in a parallel way to look at proteins, look at genes, and to cell sort. It combines those things. And it turns out it's actually pretty good. And if you control the surface chemistry on how you DNA load this chip, then your protein sensitivity is pretty much you get what, even on very short timescales like I'll talk about, you can get what the manufacturer specs for a 96-well plate, okay? But there's ways that have been developed over the past couple of years to amplify that, and one of them just uses photographic amplification, where you can get another factor of 100 or 1,000. It turns out I don't think that's a good thing to do, because it introduces noise. An additional step introduces noise. But it turns out if you do a simple ELISA assay, and you, and instead of a fluorophore, you put a little tiny gold particle here, you'd never see this by optical microscopy, but it has a great scattering cross-section. So you just put an aperture over your microscope, and you can count the proteins. You don't photo bleach and anything like that. And so this is out of human healthy blood and spiked blood. These are spiked blood. And so, so what it allows you to do is actually get down to about 100 attomolar type of concentrations of detection. And it's digital, so it's quantitative. And you get about a million-fold dynamic range in the concentration. And so for abundant proteins, you can do fluorescent assays, and I'll talk about those mostly. But you can also do this. And it gives you, it really allows you to respect the biology, this dramatic concentration range of proteins in the blood. In fact, these proteins are typically not seen in healthy humans because they're at such low levels, but this method allows you to quantitate them. So that gives us a way that scales. It's just limited by this DNA addressing. It lets us scale to lots of proteins, but it's not necessarily faster. So this is how we make it fast. So we use a trick that's in the body. And there's been, and it's called a Zweifach-Fung effect. It's well known. There's a paper you can read on this. But we've integrated with a couple of other things. And the way it works is that you've got this big fat channel that splits into a fat one and a thin one. And as this one gets fatter or that one gets thinner, there's a critical streamline that moves south. And everything with the center of mass north of that streamline heads up here. And so this allows you, this is a chip for 8 samples. It allows you to separate the plasma from the from the cells. And it works really great. It works better than centrifugation. And you'll see in this movie how it works. Here's whole human blood coming in. These are the skimmer channels, and here's the movie. And so, and there's the waste blood. About 15 or 20% of the plasma is being skimmed off. So there's no moving parts here. It's just glass and plastic. And here, and out here, you'll see it's leaking a little bit, but it's 99.999 or something like that. It's really good. And so, yeah, here you can see it's a few cells make it through, but generally it's great. But it has the other advantage that in this region, back where the blood is flowing, if you want to do your assay in that region, you're not limited by diffusion. All you care about under those conditions is the kinetics of the antibody and the protein binding. And that means you can do your entire assay in just a few minutes. And so here's an example here of taking, going from a finger prick of actually this guy's blood to a completed assay and it's just a 3 microliter finger prick, but the whole thing is completed in less than 10 minutes, really on the time scale of the blood clotting or the kinetics. And this is not with automation. With automation, it'll be down to 5. It will beat the blood clotting. And there's a lot of reasons to do this. And the most compelling one is, I'll come back to that in a second, is shown right here. So here's our assay. Here we've done a dozen proteins, and you can see the crosstalk is great. That basically these are really good antibodies. And here we've looked at melanoma patients before and after treatment, and these are stored samples. And the biggest difference is not whether they were treated or not. It's whether you centrifuge the stored plasma sample. You know, people complain because they buy antibodies and after a couple of weeks they go bad. Well, so does the blood, so does the serum and the plasma. Those proteins are denaturing. They're not going to bind in the same way. You're going to get biofouling because of denaturing, et cetera. And so it is, you know, and a year ago, the National Cancer Institute went around the country and they looked at 13,000 stored serum and plasma samples. And they just asked the simple question, which ones of these could we get a reliable measurement from? And the answer was like 100. I mean, that's staggering. I know there's a bunch of graduate students in here, and that's like at the end of your 8 years at MIT. We'll get you out faster at Caltech. But someone tells you that 99% of your dissertation work is invalid. I mean, that's what that number means. And so being able to, and so the only time we can really get truly reliable measurements is when we do this measurement within just a few minutes of taking the blood. And I'll just say that this is just some data from cancer patients. And here we're looking at organ-specific proteins, but mostly it's just the immune system plus a couple of organ-specific markers, cancer markers. And this is not an interesting stratification. We're now at 35 proteins, which actually is interesting, and that's what's in the clinic. But it's the kind of stratification that you see typically from taking a tissue sample out and measuring it. But this is non-invasive. And so the idea is that, you know, if you can, people mentioned this before, but this model of, you know, of diabetes being transformed by sampling the glucose and using it to control your diet and your exercise and all that, I mean, that's the kind of thing that one would like to be able to do for cancer. I don't know if it's possible, but it's an interesting hypothesis. And so where we are now is this is robotics that we've set up in my lab, and I've hired a guy who has an undergraduate degree to come in and run it. And we can make about 48 chips per day. Each chip does about 8 patients. We're up to, right now we're monitoring these organs. So it's on the order, when we're up to 40, it'll be 10,000 proteins. It's about 12 cents per measurement, per protein measurement. And I think we got to get down to about a penny before we really get, be where we want to be. Okay. So I'm going to run out of time, I'm sure. So I'll just mention another application of this that's more therapeutically oriented. And it has to do with, you know, I just showed you that this large amount of inflammation in these cancer patients, that was what you're looking at when I stratified those patients. It turns out that the immune system has a really striking relationship with the cancer. And on one hand, it's the most powerful weapon in terms of a therapy. And this just shows, you know, Gleevec, a famous small molecule, an antibody like Herceptin or something, and then a T lymphocyte. On the other hand, if you look at a solid tumor that's progressed, as much as half of it can be immune cells. And those immune cells are actually promoting many of the hallmarks of cancer. And so, and this is a really intriguing problem that we're beginning to look at through some of these cell sorting and molecular analysis technologies. And I'll just talk about this one and maybe give you a feeling for what we see on the other one. So the way that the immunotherapy works, when it naturally works well, is that the tumor cells, like every other cell in your body, are chopping up proteins in their internal workings and putting them on the surface as antigens inside these major histocompatibility complexes. And T cells will be sampling all these cells, and if they see something there that's not right, like it doesn't come from your DNA, then that triggers the T cell to attack, and if the tumor cell maybe have mutated proteins, this may be a bad, something foreign, and then you'll get this attack, and the T cell will kill the tumor. Now, it turns out, oftentimes the tumor cells are presenting peptides in these MHCs that should be there. And it also turns out that decoding this conversation, and there's a little tiny peptide there, and figuring out what it is like trying to drag an elephant, by the short hairs. And it's a tough challenge. But it turns out when this therapy works, it's a cure. And here's an example of a patient who the T cells were taken out, they were transfected, genetically modified so they would go back in and attack the cancer. And you see this cancer dramatically shrunk, but look at this tumor was unaffected. And that's because the conversation that the immune system and the cancer that you wanted to put in there, you only knew part of it. You didn't know all of it. And so, so you just got part of the cancer. And so the challenge is, can you find a way to do that in a multiplexed fashion, to decode that conversation? And so we took our cell sorting strategies with both human and mouse cell lines and just modified commercial antigens, commercial capture agents to these specific T cells, these basically MHC tetramers. And we cell sorted. And so it worked pretty well here. Here it worked a little bit. Here, my mother would agree it worked, but probably no one here. And this looks like a control experiment, and is not. And then my student Gabe used, we've heard a lot about cooperativity from, I think, almost every talk today. Decided to go back and really engineer this tetramer so that we really knew where all the bits and pieces were. And going on work by Altman's group, who realized that streptavidin just has no native cysteines. You can put cysteines at these corners. We can put our DNA labels away from this and make this complex here. And it turns out this works fantastically well. And these are the same exact cells you just saw, but now we're actually quantitatively sorting them. And it actually has efficiency that's as good or better than FACS, so it's better than a part in 1,000. And you can see that. And it multiplexes because we're doing DNA labeling. And so this allows us now to begin, here's in a human sample, to begin, in this case, we're addressing whether the immune system in the human is actually highlighted or alarmed against the cancer. And you find that it's actually, here's the FACS data, but with a much, much smaller amount of tissue, we can answer the same question, or blood. And you can find that there's not really much of an immune response. And so then you can take the T cells out of that patient, transfect them, and then here's our, these are cells that, except for the dye, are ready to go back into the patient to fight the cancer. And you can see the corresponding. And it's very, very clean data. And now with the technology from Tone Shoemaker's group, you can actually make a dramatic number of capture agents. And we're trying to do that to see if we can't capture the entire conversation between the tumor cells and the immune system that exists. Okay? And so in my last couple minutes, I'm just going to say a few words about this yang, this issue of the immune system promoting the cancer. And so there's this is a model, this reductionist view of cancer, but it's not like that. It's like I mentioned, it's got all kinds of stuff in it, including these immune cells. And these immune cells really are thought to promote some of these hallmarks of cancer. And trying to decode that conversation in this stroma of the tumor is almost impossible. And so my postdoc, Rong Fan, set out to just a really model system, but I'll show that we can make it reality, to just build up a tumor one cell at a time and monitor that conversation. And so this is just an example of with this experiment, 500 of these on a chip. There's one cell. We put a barcode in here, and now it's up to like 20-some proteins that we can measure from these individual cells, or 2 cells, or 3 cells. Just go through this and just go to the data. And so here's the results from his first experiment. Now we have like just a lot of data, but it's all sort of panned out to look something like this. It's really interesting. So first, when you add, this is the number of cells along this axis, and here's the barcode for the proteins that were measured. So first, if you notice one cell, this one protein here is TNF-alpha. It's digital. It's either there or it's not. And I think everybody sort of expects that. Either you turn on a process or you don't. And as you build up a colony of those cells, that protein no longer is digital. It's actually constant. Okay? But look, another protein steps in and becomes digital. And once it's constant, another one steps in and becomes digital, and then another one. And so what you find, and this is not tumor and immune cells. This is just immune cells. This is the simplest first experiment. So number one is that 43 cells is not like 44, and neither one of them is even anything close to the sum of 43 or 44 single-cell experiments. So you have this emergent phenomena, this cooperation happening, that at least seems to retain digital nature up through at least a few hundred cells from what we can tell so far. And this just shows you that experiment in graphical form. It allows you to begin putting together these communication networks, but I think there's an interesting thing that it lets you do. And so if you have a great measurement capacity and all you do is measure stuff, then that's stupid. You ought to be able to do something more with it. And so I got this idea from Bob Austin, where you can play games, or have the cells play the game, have the immune cells and the cancer cells play a game, and look at these proteins as the outcome of the game, the communication of the game. And, there's a simple game one can play where you have a partner and you each put out 1 finger or 2 fingers. This is a really dumb game. So if you're on the web, go back, okay? And then at the end of and so the result of the game is that you just add up, your finger and his finger. Maybe he put out 1, you put out 1, 2 and 1, whatever. And you add them up. And if you each put out 1 finger apiece, you get the number 2. That guy pays you $2. If you get an odd number, you pay him $3. If you get both did 2, $4, you get paid. Even number you get paid, odd number you have to pay. So you go around the loop, there's 25% chance of any one of these things coming up. You can potentially make $6 or pay $6. So can your partner. It seems like a dumb game. But it turns out you can win this game if you're the even guy. Okay? And so the point is, if you play enough of these games over time, the pressure here, the evolutionary pressure, is that you get bored. You want to make it interesting. Okay? That you'll see the results of that game change. But you put these cells in the system under evolutionary pressure, and you'll see the results of the game change. And you can actually figure out what game's being played because of things like the Nash minimum and other mathematical constructs for games. And so that's the general idea. And it seems too realistic, but now we've got, and this is just patient data from a patient 3 days ago, still alive, where we're doing this now to assign therapy, and it's actually very interesting what we're learning. But we're able to now pull apart, where we know how the game's been played out, pull apart the tumor, where we know how it's been played out, into individual cell components, and see how the conversation progressed. So the idea is there ought to be a self-consistency in these kind of experiments. And I don't know if that'll pan out, but I think it's a very interesting way to think about the science. Okay, I'm not going to talk about this. It's a big project in my group, but I think if this problem is not solved, antibodies, if you want to make money, I think you should, sell little bottles of water for $50 because or $100. That's like buying an antibody, And sometimes it doesn't work. It's really expensive. And, you need something you can put in your trunk on a hot August afternoon in Pasadena, have a kilogram of it, and come back the next year and it's still fine. And if you can't solve that reagent problem, I think this nature of diagnostics is really, that's the technological limiter in this whole game. There's a lot of collaborators here. I want to particularly thank Caius Radu and Tony Rebus, Paul Michel, Lee Hood, and Owen Whitty, and Sein Rong Sang. Mark Davis probably doesn't know it, but we use some equipment in his lab. Now he knows. And then I tried to mention or put pictures of the folks that did the work, but I circled them again here, a truly spectacular group of people, and I thank you for your attention.